Hi, folks. This is Dr. Pickett. This is the final slide narrative for Lecture 14, covering Chapter 24, our Cancer Biology / Cancer Genetics chapter. I wanted to start up again discussing slide 24. So we talked a little bit previously about genome rearrangements and building the cancer genome, the different approaches, if you will, that cancers use to evolve a genome over time that serves their needs. And we'll talk more specifically about the genes that support tumorigenesis and tumor biology in just a bit. Because of some of the very specific types of chromosomal rearrangements that have occurred in some cancers where they are stereotypically seen time after time from patient after patient, we are beginning to learn a little bit more about not only the sort of karyotype level problems that are occurring, what are the two chromosomes that are aberrantly recombined to produce a chromosome that promotes cancer, but also because now with the power of DNA sequencing and studying the human genome, we can ask a question, what is going on at the gene that might be at the breakpoint where two chromosomes recombine. And so the specific case of that I would like to discuss is one of the blood cancers. This is chronic myelogenous leukemia, or CML. What we see particularly with more late juvenile, early adult onset forms of CML is that all tumor cells have a very specific translocation. It is a reciprocal translocation. It involves an exchange between chromosome 9 and chromosome 22. And, this is further evidence of the clonality of the tumors, because wherever we look in the body where there is a tumor hotspot, if we do a needle biopsy or do a liquid biopsy for circulating tumor cells, we discover that in CML patients, there is some heterogeneity, but we won't really go into that too much in the context of this course. But in the vast majority of patients that we'll be looking at clinically, we see a rearrangement, a translocation of chromosome 9 and 22. Now, the derived chromosome, the translocation chromosome from these particular cells has come to be referred to as the Philadelphia chromosome. And so if you'll refer to slide 25, you can see on the left side a comparison of two normal non-homologous chromosomes, one copy of chromosome 9 and one normal copy of chromosome 2, showing the potential, what we would refer to as translocation breakpoint, the site at which the aberrant recombination had occurred. We see two sites. One is on the chromosome 9 copy, the other is on the chromosome 2 copy. What are the two genes that we find there? One is the Proto Oncogene on chromosome 9. The other is the BCR gene, which is present on chromosome 22. When there is a non-homologous exchange or recombination, a translocation between these two chromosomes, this produces two derived chromosomes, one called T922, in which the terminal region of the Q arm of 22 has been translocated onto chromosome 9. Its role in leukemia or other disease is pretty much excluded at this point. We don't think this translocation is strongly associated with the disease state that these patients experience. However, the other chromosome, the first one was T22-9, The second one is T922. This produces the very short derived version with the centromere of chromosome 22. If we kind of moved along the DNA from the centromere of chromosome 22 to this translocated telomere, what would we find? Well, as we were moving from the centromere, we would see chromosome 22 information. Then we would reach the translocation breakpoint, and from that point on to the telomere, we would see exclusively chromosome 9 information. So this is a real translocation. It is reciprocal. A bit of chromosome 22 has been translocated onto 9. A bit of 9 has been translocated onto chromosome 22. What is important is that the Abelson kinase has now been converted by fusion with another gene, the BCR gene, into a kinase. And remember, kinase phosphorylates target molecules, has been converted into a kinase that is now associated with activating proteins and other components. We think also cytoskeletal elements that appear to promote proliferative cell division in derivative cells from the hematopoietic stem cell lineage. So this is why we end up with a blood cancer, why we end up with a leukemia. These cells are not able to go ahead and fully differentiate as appropriate forms of white blood cells, and instead continue to grow and develop as these kind of intermediate cancer cells. So this translocation is seen almost universally, not exclusively universally, particularly amongst infant and very early juvenile forms of this disease. You may see other types of problems popping up, but certainly in juvenile and later forms of this disease, whenever you sample somebody's tumor, if we karyotype or if we do a sequencing experiment of those tumors, we find the Philadelphia chromosome, as seen on slide 25. So this is quite a fascinating example where an illegitimate exchange of DNA creates now a fusion gene that loses some of its normal regulation, but still, following transcription and translation and splicing, produces a protein that has lost some normal function, but you would argue perhaps gained other function through the loss of that activity, becoming an unregulated kinase that starts activating lots of proteins that cause other problems in the cell, in particular proliferation and growth. So what are some of the actual genes that we see that are involved in oncogenesis? We just talked about the Abelson oncogene, and we'll talk a little bit about what that means in just a bit. But we definitely see cancers associated with mutations in genes, whether they are loss of function, amorphic or hypomorphic alleles, or coupled loss and gain of function alleles like the Abelson oncogene, or gain of function alleles, as we see with some forms of our guardian of the genome p53 mutations. So in 24.2, basically on slide 26, what we're basically discussing are a couple of the associated mutations that we see in genes associated with specific cancer syndromes in patients. Xeroderma pigmentosum. What do we see? We see defects in genes that are involved in DNA repair. Hereditary nonpolyposis colorectal cancer. HNPCC. This is quite fascinating because although this is sort of one characteristic syndrome, we see that distinct mutations in 8 different genes can leave you with a hereditary nonpolyposis colorectal cancer type of phenotype. 4 of those are involved in DNA mismatch repair when there is a situation where perhaps you have a G and a T, a G in the nascent strand of DNA, a T in the template strand of DNA. If you don't have good mismatch repair, if that helix is stable and goes through subsequent DNA replication, you will immortalize in the daughter cell a new mutant allele of one of these genes. And so mismatch repair, mutations in mismatch repair is a very powerful cancer mutator phenotype. If you are corrupting the DNA and you make corruption more common across the genome, more and more mutations will accumulate in different genes where loss of function, partial loss of function, or gain of function mutations promote cell division and oncogenesis. I think I'd like to skip through to slide 29. We've chatted a little bit about epigenetics previously, but certainly methylation, modification of histones can have an impact on gene regulation, which as well can contribute to mutator or other tumor phenotypes. With 29, we begin to think a little bit more about cell biology and how mutation can reach in to the core cell biology of the cell and alter some of the fully natural, fully normal processes of cellular activity, physiology, and biochemistry, and hijack them towards promoting a cancer phenotype. Not surprisingly, what do we discover is mutations in genes that are regulating the cell cycle, that cause defects in cell cycle regulation. Now, moving to slide 30, as we've discussed previously, obviously growth and differentiation of cells is tightly regulated. Now, what do we mean by growth? We remember previously we talked about growth from a cellular perspective, i.e., cells increasing in size through acquisition of water, increase in biomass through the synthesis of proteins, DNA, etc. But then growth also has sort of a proliferation phenomena where growth from the perspective of an organ or growth from the perspective of a tumor will also incorporate cell division. Not only will cells be increasing in size, will the amount of protein per gram of tissue be increasing, but also cell counts will be increasing and the number and type of cells may change over time. So oftentimes you'll remember that there are many genes that regulate the cell division cycle as we transition around the nice little clock going from G1 to S to G2 to M, moving between G1 into G0 quiescence. All of these processes are quite tightly regulated by gene expression. Different genes are correlated with different stages. Genes in particular, and their expression is in particular, associated with transitions between, for instance, G1 and S, S and G2, and G2 and M. We call the genes that are assessing these transitions between different parts of the cell division cycle cell division cycle checkpoint genes. They've often been called CDC, cell division cycle genes. If we look at the yeast literature, CDKs, cell division kinases. If we have a CD as part of our gene name, chances are good cell division is part of its role in the cell. Mutations in many of these genes, loss of function, gain of function, amorphy, hypomorphy, neomorphy, hypermorphy. These particular allele types, if they influence genes that are playing an important role in regulating the cell division cycle can definitely lead to cell proliferation and cancer. Unregulated cell division is at the core of tumorigenesis. All right, so let's transition to 31. Now, I know we've discussed it in depth quite a bit previously to this point, but just as a reminder, two daughter cells exiting the cell division cycle arising from a single mother cell will transition from M into G1. During G1, what is occurring? Transcription, translation, accumulation of biomass, biosynthesis, cellular physiology. A lot of the business keeping of the cell may be happening in G1. Cells stably placed in tissues that are receiving cytokine signals or adhesion signals through interacting with other cells may exit G1 into G0 or quiescence, and they will, in effect, be opting out of further division at that point unless there is a local change in the tissue. These cells may go from quiescence to senescence quite effectively. So G0 can be a gateway to simply a cell in skin biding its time until finally it's shed as a dead skin cell. So cells, if they do stop proliferating, they will enter G0. They are no longer dividing, but again, they are metabolically active. A classic example of that is neurons. You may have many, many, many very old neurons in your brain. And, you know, the ability of the brain to regenerate after damage is interesting, controversial. We now have found really good adult neural stem cells, which are available to repair some types of damage in the brain if they're lost to something like stroke or physical injury. But neurons are thought of as kind of, if you will, the quintessential G0 cell. They're just hanging out being neurons. They're not going to divide further over time, and only significant injury in the area may recruit neural stem cells to come in and re-differentiate as neurons, but the neurons themselves that are present in that region will never normally back up and become stem cell-like and undergo division. One thing we see with cancer cells is that one of the things they appear to lose very early is their ability to transition from G1 into G0. They literally do not have the ability to go quiescent. And two types of therapies that are out there now. One is called differentiation therapy. This is, in fact, very common in various types of leukemias like CML, AML, etc. We have a tendency to treat these patients with very strong developmental drugs that are trying to get their myeloid lineage or other hematopoietic lineages to take cancer cells from a cancer identity into a more terminal identity as a white blood cell. Why? Because if they do that, those cells enter quiescence and they no longer keep dividing. So quiescence therapy is this idea of, might we find a regulated way to move dividing cells into G0? So even if they're cancer cells with cancer genomes, we might be able to trick them into thinking, oh, I'm part of a tissue, rather than being a liver tumor of less differentiated, simpler, earlier in development looking cells. I might be able to get a little cluster of hepatocytes to form, or maybe I'll be able to trans-differentiate them into something like a little cluster of skin. Well, it might not be great to have little blobs of skin growing in your liver, but that might be treatable by surgery or might be treatable by other types of approaches where letting a hepatocarcinoma take over the body simply would not work. So G0 is an attractive, very seductive research target for us. It would be wonderful if we could drive cancer cells that are just cranking through the cell division cycle, mothers making daughters, daughters becoming mothers, making daughters, becoming mothers, making daughters. If we could get those cells into G0, we might be well on our way to trying to beat cancer. So let's transition to slide 33 and talk a little bit about these different cell division cycle checkpoints. Three that are discussed in particular in your book are the checkpoint between G1 and S, synthesis. The second is a checkpoint between G2 and M. That's a very popular checkpoint for drug development, trying to study the genes that are playing part of that role, and also mitotic checkpoints. So in slide 34, there's figure 24.5, which provides a nice schematic of the normal cell division cycle, which we've discussed previously in class, M leading to G1, G0, quiescence being an opportunity available to the cell in G1, G1 moving to S, S moving to G2, and then G2 moving to M. But you'll notice with the arrows and text boxes that we've added in labels of the G1 to S checkpoint, the G2 to M checkpoint, and then finally the mitosis checkpoint as well. So what's going on at these different checkpoints? What is the biochemical machinery of the cell assessing at G1S or assessing at G2M? Well, if we look at G1S, we believe that cell size, if you will, biomass, accumulation of biochemical products, but also the integrity of the DNA, presence of mismatches, presence of breaks and other types of problems is monitored at the G1S checkpoint. Remember that after S and then entering into G2, we will have completed DNA replication. So the G2M checkpoint is quite fascinating because it is assessing, in effect, the fidelity of DNA synthesis. Has the sequence of DNA nucleotides in the mother helices been faithfully transferred through DNA replication to the two daughter helices present in the two chromatids of a single DNA homolog? So the cell at this point is monitoring the quality of DNA synthesis. Did we have good base pair complementarity? Is the DNA that came out of S phase as good as the mother helix that went into it? M checkpoint, what do we think we're looking at here? Well, remember, by the time we go through S phase, we now have homologs, and those homologous chromosomes each have two chromatids on them. The chromosomes will, if you'll remember, attach to the spindle apparatus through the kinetochore proteins that are associated with the centromere. And if they are mounted appropriately to the spindle apparatus, eventually the sister chromatids will separate, undergoing the transition, which I describe as the tid to zome transition. The two sister chromatids will become chromosomes, and they will then segregate away from each other towards the spindle poles in the mother cell, where they will become the genomes of the two daughter cells. So we think the M checkpoint is a lot about assessing, has the spindle attached appropriately? Do we have appropriate metaphase placement? Has there been good kinetochore capture by the spindle? If that's true, that means we've got the machinery of mitosis all lined up, ready for the separation of the sister chromatids to then haul those new chromosomes apart to make sure that we will take the euploid mother cell and create euploid daughter cells. If we don't have good spindle attachment, what's going to happen? We're going to have chromosomes lost, left behind at the metaphase plate. So that will produce daughter cells with unbalanced genomes, daughter cells that are monosomic potentially, or trisomic for some of the chromosomes, using the language we talked about when we discussed macromutations. So now let's talk a little bit about the regulation that is involved in assessing whether or not cells should transition through these checkpoints. Regulation of cell cycle progress is mediated by two classes of proteins, cyclins, which are regulatory proteins, and they play a very important role through interacting with their partner, the cyclin-dependent kinases. So we call this the cyclin CDK cycle. And one of the reasons we'll call these proteins cyclins is we'll see that transcription and translation of these proteins is kind of timed during the cell division cycle. So at some points in the cycle, some cyclins are increasing in concentration, while at that same point in the cycle, other proteins might be decreasing in concentration. So since the cyclins rise and fall in concentration as the cell cycle progresses, this is the source of their name as cyclins. Thus, the famous quote, cyclins cycle. They increase in concentration and then decrease in concentration, and that correlates with progression through different parts of the cell cycle. So if we transition to figure 36, we see a very nice discussion or description of the relative abundance of the different cyclin proteins at different phases of the cell cycle. So here on the left axis, what we see is the relative amounts of cyclin. So if you're higher up the graph, we are detecting a higher concentration of the proteins in cells, cell cytoplasm derived from cells at that stage. Along the lower axis, what do we see? We see the cell cycle mapped out over time, G1 adjacent to S, adjacent to G2, and finally M on the farthest to the right of that axis. So here what we're able to do is we're able to chart the relative concentration of cyclins with cell division time as our time scale. So for instance, what do we see? We see that cyclin D1 increases with concentration beginning with G1, then holds quite steady until right at the end of mitosis when its concentration drops off. We see if we were to give a cyclin address to something like the G1S transition, well, the cell cytoplasm knows it's in G1S because D2 is high, cyclin D1 is at a fairly constant concentration, cyclin E is decreasing in concentration, and cyclin A is increasing. We see the G2M transition is quite dynamic. How do we know we're in G2M? Well, D1 concentration is steady, then begins to drop, but cyclin A and cyclin B concentration peaks. So the abundance and representation of different cyclins in these cells gives a molecular address in time to the cytoplasm of cells in the cell division cycle. A cell knows, if you will, what time it is based on the relative abundance of the cyclins that are there. Now, we'll talk just a bit here about the biochemistry of the cyclins and how they work. And so we'll understand what it means that these cyclins are passing a threshold required for concentration, or threshold concentration required for activity. Once they exceed that threshold, their local concentration is high enough that the job they do is done. But then as they are degraded, they will eventually drop below that threshold. And when they do, their job is no longer done. So what are we seeing here? We're seeing jobs being done, then being turned off, jobs being done, then being turned off. A successive wave of jobs leading the cell, or activities, leading the cell through the cell division cycle. So in slide 37, we have a nice example of the function of Cyclin B, the regulatory protein, and CDK1, its partner kinase, at the G2M transition. Remember, I told you from the previous graph, Cyclin B concentration is peaking as we get to G2M, then it will quickly drop off as we transition into mitosis, excuse me. So what is that job? Okay, so Cyclin B's transcription and translation increases. Other stability factors have an effect. Cyclin B's concentration increases. It passes its threshold, and what does it do? It binds to cyclin-dependent kinase 1. By binding to cyclin-dependent kinase 1, it activates its function. This allows cyclin kinase 1 to begin to phosphorylate its target metaphase proteins. By activating that phosphorylation and by post-translationally modifying the target proteins, the cell begins to be locked into mitosis. If that concentration of B doesn't get high enough to activate CDK1, those proteins are not phosphorylated and mitosis does not continue. We describe that as a checkpoint arrest. Now, once we get into M phase, what begins to happen? Specific protease activities begin to degrade cyclin B. Cyclin B concentration sinks in the cell. Less cyclin B is available to bind to CDK1. CDK1 turns off. It's no longer phosphorylating its targets, and we exit mitosis. And once anaphase is complete, telophase is complete, and we begin to re-enter G1 through the process of cell division. We've gone from a mother cell to two daughter cells, and the two daughter cells will then re-enter the cell division cycle. So what are some of the signals that we think for helping a cell undergo a checkpoint arrest? What causes cells to take a pause and go, DNA replication didn't go too well, or, we haven't really synthesized enough of the proteins we need to do good DNA replication. As a cell, I'm not confident. I hate to anthropomorphize, I'm sorry. I'm not confident in moving on into the cell division cycle. I'm not sure I've got enough of the stuff I need to complete this job. So cells tend to halt their progress if they are not detecting, for instance, presence and activity of enough of the synthetic enzymes, if there is a lot of DNA damage, if DNA replication and repair is aberrant, if chromosome assembly, if folding a nice metaphase chromosome into a nice metaphase conformation has not occurred through appropriate chromatin condensation, then we're going to pause the cell cycle and we're going to try to mobilize factors to fix it. Now, in situations where the damage, particularly DNA damage, is so severe that repair ends up becoming impossible, it is quite fascinating because the cells will usually elect suicide. The factors that regulate apoptosis, programmed cell death, will accumulate to a high enough concentration. They will pass their threshold of activity required, and once they do, they will ruthlessly drive the cell into suicide. They will cause the destruction of the mitochondria, the destruction of the nucleus, and the cell will apoptose, undergo programmed cell death. So this is a very important thing if we think about cancer. Cells with aberrant DNA may be on the path of developing a mutator genotype and mutator phenotype. If apoptosis happens, destroying that cell means it is no longer available to eventually contribute to cancer. So a way, if you will, to break the cancer lineage would be to induce apoptosis in cells with aberrant mutated genes. If we could just drive the cell into apoptosis, that would be quite helpful. So it's interesting because apoptosis appears to play a very important role in making sure that the mother cell genome is replicated with high fidelity and distributed with high fidelity to the daughter cells. And if that does not occur, to kill the cell that is having problems. But interestingly, programmed cell death also occurs as a normal process in development. So, there's some controversy about the evolution of programmed cell death. And there's a little bit of a chicken and the egg question. Did programmed cell death arise with multicellularity? Well, some of the genes are, well, certainly the cyclins, et cetera, but also, the evolution of the genes that regulate apoptosis is fascinating. And are there ancient genes that have some of the same characteristics? And so things are very interesting. It is likely, I would say, that apoptosis did predate multicellularity, but it really became important in multicellularity when organisms had to organize tissues, organize coherent structures, and develop them appropriately over time. The famous nematode, Caenorhabditis elegans, has many many programmed cell death genes, PCD genes, that are involved in shaping the final form of the mature organism. So in order to get a normal worm out of embryogenesis, some cells must die. If those cells don't die, you end up with an aberrant phenotype, aberrant physiology, and other problems. So apoptosis and programmed cell death plays a very important role in anti-neoplasia, anti-cancer aspects, but also plays an important role in development. In slide 39, we talk a little bit about two of the key genes that play an important role in regulating apoptosis. And so I'd like to just discuss them briefly. These are very closely related to each other. These are the BCL2 and BAX proteins and genes. They can make homodimers with each other. So BCL2 copies can bind to BCL2 copies, two blue Legos sticking together. BAX also can make homodimers, two red Legos sticking together. But interestingly, these Legos can also stick to each other. So we find them in homodimeric and heterodimeric forms in cells. What do these different forms mean? So if the concentration of BCL2 is very, very high, most BCL2 will be interacting with other BCL2. You'll have a lot of BCL2 homodimers. If the concentration of BAX is very, very high, most BAX homodimers, most BAX subunits will homodimerize with each other, and you'll have a BAX homodimer. However, if the concentrations are kind of intermediate, BCL2 and BAX will bind together and you'll have a heterodimer. So what do these different machines do? It's quite fascinating. If you have a BCL2 homodimer, you actually have a protein that is active in blocking apoptosis. So if the BCL2 protein accumulates at a high level, especially in relation to BAX1 concentration, which is low, the cell will be protected from apoptosis and will be permitted to progress through the cell cycle. If you have BCL2 and BAX together, it's interesting. You're neither protecting against apoptosis nor promoting it. They're kind of canceling each other's activity out. But if you have a Bax-Bax homodimer, now you have an activator that promotes apoptosis, turns on PCD, programmed cell death genes, and will begin to drive the activity of caspases. These are important proteins that play a role in cell death. So slide 40 is fun because it kind of really shows you two characteristic cells. These are scanning electron micrographs of a normal tissue culture cell and a tissue culture cell in which you have generated Bax-Bax homodimers. So the tissue culture cell that is on top is undergoing programmed cell death. The tissue culture on the bottom is a normal cell. Based on their approach to preparation of these samples, you see with the normal cell, it's nice and spherical. It's got a few little membrane projections sticking out the side. It's a nice little kind of beach ball floating in space. But the apoptotic cell, what do we see? Looks a bit like a cauliflower, doesn't it? It's got these little blobs and blebs and nubbins kind of sticking out. What is that? Well, as the cell is undergoing death, little parts of the nucleus are beginning to be enfolded by little outwellings of the membrane. Eventually, this cell will begin falling apart, and macrophages and other cells in the human body will digest that cell and destroy it. So this is a very characteristic cellular phenotype of a cell undergoing programmed cell death. Why is that important? That is in contradistinction to a cell that is undergoing necrosis or crush damage leading to death, etc. Programmed cell death is a specific regulated phenotype of cells that is distinct from the other ways we believe. Now, there's a little bit of controversy, which I won't get into in the context of this class, but it's quite a distinct phenotype in relation to garden variety death of a cell. So in 42, we're kind of discussing the steps that are occurring in apoptosis when we have a situation where we've got high numbers of Bax-Bax homodimers. We see fragmentation of the nuclear envelope. You begin to get degradation of DNA, the destruction of chromosomes. So as the nuclear envelope is blebbing off, it's taking little chunks of chromosome with it. We're beginning to slowly chew up all the intracellular compartments of the cell. You have disruption of internal cellular structures. We begin to lose Golgi. We begin to lose the ER. You end up finally with dissolution of the cell into these small spherical apoptotic bodies. You kind of think they undock from the cell, taking bits of cytoplasm, bits of nucleus, bits of DNA with them. And eventually these small phagocytic or apoptotic bodies are phagocytosed. They are chewed up by phagocytic cells, many of them white blood cells. What are some of the proteins that are either directly or indirectly regulated by Bax homodimers? Those are the caspases and other proteases that are important for initiating apoptosis and digesting intracellular components. Okay, so at this point, let's chat a little bit more about some of these genes that are mutated. So I'd like to refer you to slide 44, section 24.4, where we'll begin to discuss Proto-Oncogenes, oncogenes, and tumor suppressor genes, and we'll define those formally. So let's start with the Proto-Oncogene. They do give you a nice table in table 24.1, which I would refer you to, that kind of lists different well-known, well-characterized proto-oncogenes, discusses their oncogenic mutated form, and then other genes, which are tumor suppressor genes, and also talks about their alleles, which result in loss of tumor suppression. So to begin with, what is a proto-oncogene? So these are genes whose wild type alleles produce products that promote cell growth and division. So what might they include? Transcription factors that regulate the expression of other genes, cyclins, etc., other targets like that. There are also signal transduction molecules, i.e. receptors, ligands, signaling intermediates along the signal transduction cascade that are involved in stimulating cell division. They are also, obviously, the cell cycle regulators that move through the cell cycle. So oftentimes what we see in a cancer genome is that proto-oncogenes have been converted from their normal wild type cellular allele into an allele that is causing a simultaneous loss and gain of function. It causes a loss of the normal regulation of that protein, but as that regulation is lost, the resulting protein, the other domains in the protein, by being unregulated, become constitutively active. So you might think of this as equivalent if you think about, a light switch. A light switch switches on between off when the circuit is incomplete and on when the circuit is complete. But if I pull the light switch out of the wall and just twist the wires together, the lights are always on, the circuit is always complete because we've lost regulation. That's analogous to what happens as proto-oncogenes through mutation become oncogenic alleles. We are destroying regulation but leaving some function there. We've cut out the switch, but by putting the wires together, we have made an unregulated but continuously active circuit. So what do we see? By causing these proto-oncogene to oncogene mutations, we've converted a protein into a protein, usually that is continuously promoting cell division, loss of cell adhesion, change in cell type. It's driving cancer forward when proto-oncogenes become oncogenes. Let's contrast that with tumor suppressor genes. Tumor suppressor genes are interesting. If we go back to our light switch analogy, tumor suppressor genes would be a thumb holding the light switch off. And so tumor suppressor wild type genes play a role keeping things turned off, keeping activities from happening. So the problem there is if a switch is flipped on and we cut off the thumb, we're stuck with a light switch that's always on. It's constitutively active. So tumor suppressor genes play a suppressor role. They're like, let's pause cell division and see how DNA is doing. Let's pause cell division and see what our biochemical complement of proteins is and our growth level is, our biomass is. So tumor suppressor genes play an important role pausing genes. And so their loss of function mutations often result that once something turns on, it can no longer be turned off. So if we look at slide 46, we've got two different subcategories of genes in one table. Proto-oncogenes, they talk about c-Myc, c-Kit, E6, the cyclins. The cyclins are notorious proto-oncogenes. Why? Well, imagine if cyclin B lost the ability to be degraded. It would always be present. It would always be driving cells past the G2N checkpoint. Not good. So cyclins are notorious proto-oncogenes. My lab, we work on retinoic acid and retinoic acid receptor, so RAR alpha. This is a classic proto-oncogene in its normal form. It's a retinoic acid receptor. in its mutated form, it can cause acute promyelocytic leukemia. Tumor suppressors, the retinoblastoma protein we talked about, APC, Anematous polyposis coli. P53, P53 is tough. We'll talk a little bit about that later, and you'll discuss it more if you take Father Breslin's cancer biology class. But P53 we know as a loss of function gene. It does play a role as a tumor suppressor, but unfortunately some of those loss of function alleles appear to simultaneously gain other activities. So P53 is tough because if we lose its normal activity, that is detrimental to the cell, but the same mutations that cause a loss of activity may cause a gain of function as well. So we might be losing the ability to prevent enhanced cell division, but then also simultaneously gaining a little bit like an oncogene, the ability to promote tumors. BRCA1 and BRCA2, they are both involved in DNA repair. They're very famous genes involved in breast and ovarian cancer risks. Okay, so that kind of introduces us to oncogenes and tumor suppressor genes. Let's talk about a few specific examples. The Rous gene is a famous, if you will, gene that plays an important role in regulating cell division. Normally, the Rous gene acts as part of the signal transduction cascade, as indicated here in slide 50, where you have an external growth factor that interacts with its receptor. Its receptor interacts with something called a Gdp Gtp Exchange Factor, and this is just good old nucleotide GDP or GDP. Rous plays an important role as a small, they call it GTPase. Rous in its active form is bound to GTP, and in that form it interacts with a whole kinase signal transduction cascade, eventually regulating transcription factors which play a role in promoting normal cell division. Rous mutations that cause a loss of the ability of Rous to be switched from being bound to GTP to being bound to GDP result in locking that Rous in its GTP-bound form. Thus, it is driving phosphorylation after phosphorylation, and that is promoting cell division after cell division. So this is a classic example of a Proto-Oncogene being converted into its oncogenic form through a mutation. It is interesting because these are kind of considered partial loss of function as well as gain of function mutation. It loses the ability for GTP to be exchanged with GDP, but with its GTP-bound form always present, it is driving cell division onward. P53, which we talk a little bit about here, is quite fascinating. If we refer to slide 53, what do we see? P53 plays an incredibly important role as a transcription factor. And if we look at the transcriptional targets that it's regulating, we'll see why. If we move to the bottom here in figure 2410, what do we see the genes that it's regulating? Oh, it's regulating apoptosis through activating BAX. It regulates G1S through regulating the transcription of a target protein, and which then has an activity on the cyclin CDK complexes called P21. It has a regulation through GAD45 through DNA repair. And then it also regulates its own regulator, the transcription of a gene called MDM2, which in turn plays a role in p53 control. Now, because p53 is reaching its hands in, it's an incredibly social protein. It's regulating the transcription of many target genes, but it's not surprising then that it itself is under a lot of regulation. A protein that binds directly to it is called MDM2, which plays an important role in degrading p53. So p53 concentration is always kind of oscillating in real time. It will be increasing, then decreasing. Thus, the transcription it controls increases and decreases. It's kind of held at atomic level. If apoptosis is occurring, p53-driven expression of BAX will increase. But p53, what's activating it? Things like DNA breakage, other types of problems are stabilizing p53. Its concentration is very dynamic, though, because it is under regulated protein degradation. If MDM2 binds to it, it acts as to help polyubiquitinate the protein, which then drives it towards protein degradation. So if you are an unstressed cell, p53 will drive the transcription of MDM2. MDM2 concentration will increase. It will bind to p53, and p53 will be destroyed. Thus, its concentration will drop over time. If there's DNA damage or inflammation or other types of cell stress, what do we see? MDM2 concentration is low. p53 concentration is stabilized through phosphorylation with other target kinases and other types of post-translational modification. So if DNA damage is increasing, p53 concentration is increasing. Slowly over time, what will happen? DNA repair will be activated, but if it is not successful, the concentration of p53 will rise so high that BAX concentration will increase, BAX homodimers will form, and the cell will be destroyed. So because of p53's central role and its sensitivity to things like DNA damage and the state of DNA repair, we often refer to p53 as the guardian of the genome. You'll certainly talk about that a lot more if you have Dr. Breslin's class. Now, one thing I would like to say about p53 is because of its important role, p53 and interacting proteins have long been targets for rational drug design, chemotherapy development, etc. MDM2 regulators and regulators of the MDM2 p53 interaction have turned out to be fruitful targets for chemotherapy drug development. If we can regulate how proteins interact with p53, if we prevent a p53 destroyer from interacting with it, we might be able to reactivate p53 concentrations and drive cells towards apoptosis. So that would be great. But, cancer cells are influencing all of these mutations in p53, maybe accumulating. One of the best mutations from a tumor perspective is one in which the binding site for MDM2 is lost. Thus, p53 is apparently playing a role, kind of driving cell division forward without moving it into apoptosis. It's quite fascinating. So the other two genes that we talk about in the context of this course, the Retinoblastoma Protein, which is quite fascinating, and its role in regulating the G1S checkpoint. These are also very, very well characterized online. Your book has kind of stopped emphasizing some of these different systems. I'm not sure why. But I think at this point, I'll go ahead and just leave it at that point, that many of these proto-oncogenes, their mutant form, which are oncogenic alleles, and the tumor suppressor genes are playing an important role through interacting with other proteins, influencing other proteins' activity or stability. And as mutant forms, what do they end up doing? They end up removing the light switch, wiring the wires together. What does that circuit complete? More and more cell division. Keep dividing, keep growing. They may also contribute to the mutator phenotype. Without good accumulation of DNA repair enzymes, more mutations accumulate, a classic mutator phenotype. More proto-oncogenes are converted to oncogenes. More tumor suppressor genes are lost. It's quite fascinating. So I think for the remainder of the slides, what I'd like to encourage you to do is revisit some of the text in your book for a discussion. Some of them are quite fascinating. The one I would like to talk about is one of the rare inherited cancers or more rare inherited cancers. So I'd like us to skip ahead to slide 70 to discuss the Apc gene and familial adenomatous polyposis coli, which I've already introduced briefly in the last slide narrative. So if we skip ahead to slide 70, what do we see with Apc? Apc is quite a fascinating protein. One of my colleagues described Apc as beta-catenin's bubblegum. So beta-catenin or beta-catenin, okay, pick your poison, is a fascinating molecule because it can function both part of the cellular cytoplasmic architecture, but it can also function as a transcription factor. The Apc protein binds to a lot of the proteins that regulate whether or not beta-catenin stays in the cell interacting with the cytoskeleton. It interacts particularly with the actin cytoskeleton, or is destroyed within the cytoplasm if it does accumulate it, or accumulates in the cytoplasm, but not in interaction with the actin cytoskeleton. It translocates in the nucleus where it regulates target gene expression and can promote a tumor phenotype. So in the absence of Apc, what do we tend to see? With the loss of Apc, this beta-catenin protein, which is normally targeted for destruction, accumulates at a high concentration. It moves into the nucleus where it then causes rapid cell division and the accumulation of a tumor-like phenotype. So why is Apc so fascinating? It's a fairly large gene, and there are a not insignificant number of natural loss of function alleles that are present in families. It ends up causing a dominant cancer phenotype because one wild type allele and trans to a mutant allele of Apc often goes through a phenomena called loss of heterozygosity, leaving you with only the tumor suppressor loss of function allele form of Apc. So this is a fascinating way where heterozygotes for Apc can be followed over the course of their life. These are plus over minus heterozygotes. And what do we see over time in epithelial cells or endothelial cells lining the lumen of the gut? We see first, second mutations accumulate in the one good copy of Apc. This leads to a mutator phenotype, more and more mutations in other proto-oncogenes being converted into oncogenes, loss of tumor suppressors occur. So this is an amazing natural situation to study the natural history of cancer. What are the tumor mutations that need to occur over time as we move from being a wild type cell, heterozygous for the loss of a tumor suppressor gene, to early stages of benign, non-metastatic tumor cells, but then slowly moving into malignant cells, then moving from a localized adenoma or cancerous polyp to an adenocarcinoma or other type of more severe systemic cancer that will be found all over the body. So directing you to slide 72, it's quite fascinating because here we are looking at a diagram of the natural history of this cancer, where a patient that is a heterozygote will have a normal colon epithelium. Then as some individual colon cells within the lining of the colon end up becoming essentially homozygous recessive for the loss of APC, what do we see? The epithelium starts proliferating. We start getting cellular hypertrophy, an increase in cell number. This ends up giving us a benign adenoma, which if we remove it, this is one of the reasons why we do colon cancer screening by colonoscopy. We can easily remove these adenomas. That fully resolves the cancer risk for that benign tumor. Now, other epithelia in the colon may become cancerous as they lose their APC copy, but colonoscopy with colectomy, or with a polyectomy. I'm sorry, I think I'm missing my terms there. With the removal of a polyp or removal of a small adenoma is often definitive for resolving disease in the patient. However, if there are, as we've now created proliferation, we've begun to form a mutator phenotype. What do we see later on? Ooh, Proto-Oncogenic Rous becomes mutated into oncogenic Rous. So now we've got a cell that's APC minus minus and has a Rous oncogene. Those cells end up proliferating and they produce what are referred to as intermediate adenomas, these kind of proliferating mass of cells. What do we then see? Loss of a tumor suppressor gene, DCC, correlating with the tumor phenotype, late adenoma with accessory branched villi. But then what do we finally see? Okay, we had a cell that started out as an APC loss of function heterozygote, then became APC homozygous loss of function, then APC homozygous loss of function plus a RAS mutation, homozygous DCC minus, then you get heterozygous loss of P53. Now, because that's usually correlated with a suppression in the accumulation of the wild type copy, heterozygous loss of P53 can be sufficient to drive you in. By the time you get to that, you have a cancerous adenoma. The next step is adenocarcinoma, systemic cancer, metastasis. The patient dies of their tumor cancer bleeding. So transitioning to 73, I would just like to talk a little bit about the fact that we do see some evidence that one of the insults to cells that can cause cancer is viral infection. This is why, for instance, human papillomavirus vaccination is so incredibly important to be protective against uterine, ovarian, potentially, and other Mullerian duct cancers. There's some evidence that they're protective against cancers of the oral epithelia as well, because sexual transmission of HPV can result in local infection in diverse sites in the body based on human sexual behavior. So viruses do play a role because they infect cells and they may cause proliferation, cell division, a loss of normal regulation. Now, that's in contradistinction to what we see in some animals where there are acute cancer-transforming viruses, often acute transforming retroviruses. One of the famous ones is the Rous sarcoma virus, which is discussed in slide 75, where a cellular oncogene has been captured by a virus through illegitimate recombination and then has been converted from its pro-oncogenic form into its oncogenic form through the accumulation of mutations. So this is a much more direct form of infectious cancer than we see with something like cancers associated with HPV. HPV is not an acute transforming retrovirus where a oncogene has been brought in by infection with a virus. Instead, these are not acute transforming viruses. They are simply viruses that cause misregulation of cell division. So to date, no acute transforming retroviruses have been identified in man, but cancers have been associated with many viral infections. HIV is famously associated with Kaposi's sarcoma and other types of soft tissue, bone, brain, other types of cancers. The human T-cell leukemia virus is famous following infection causing leukemia. So these are not acute transforming, but because of their activity, they do play an important role in oncogenesis. We also know that many environmental agents play a role in human cancer risk. Why? Let's talk about tobacco smoking or booth tanning or tanning through sun exposure. Why are these oncogenic? Because they modify DNA. By mutating DNA, they change Proto-Oncogenes into oncogenes. They cause the loss of tumor suppressors. So anything that is mutagenic is likely transformational in terms of cancer. Why does cigarette smoke? Why is it correlated with such an increased risk of lung cancer? Because the lungs are directly bathed in mutagenic agents that do things like methylate DNA, changing DNA sequence, causing proto-oncogenes to become oncogenes, and tumor suppressors to be lost. So anyway, at this point, I would like to bring this slide narrative to an end, and thank you very much for participating in my course. I'm sorry because of all the various disruptions we had this year that I didn't get to all of this material for these 4 little mini lectures during the course of the course, but I hope this is helpful to you in your studies. I would like to remind you, of course, that I'm available for advising. Please send me a request over Zoom if you would like to ever have a chat. I am in town all summer, off and on. I do have a few things I have to do where I might be unavailable, but I should be around most of the summer. Please consider me for future courses. You guys were quite resilient and did very well. I don't teach other than 282 in spring this year, but I would like to alert you to Bio 380, which is Evolution and Genetics of Development. I usually teach this course as an evolutionary medicine course where we take a developmental syndrome in man and examine the mouse models or the Drosophila models or Xenopus laevis or zebrafish models of that disease, and then understand how evolution informs our medical practice when we're thinking about human developmental and other types of anomalies. I become particularly interested in cardiac anomalies. We think there are many cardiac diseases that are traceable to mutations in human genes that can be studied, for instance, in zebrafish and mouse. That this year will be offered as a special JTerm course next January. So please consider joining me for that class. It's been very, very well reviewed. Previously, it's taught as kind of a combination of an activities course. We'll do things like build some models using good old Play-Doh. It'll be interesting to see how we do this online, but I think Play-Doh is available the world around. We build some models of embryos. We take those embryos through embryogenesis. We evaluate how genes are playing a really important role in early embryogenesis, and then we transition into, but what happens when these genes are mutated in man or in mice or in fish, and how do these different interactions inform ultimately our clinical practice, which is quite fascinating. So I hope you'll consider taking Bio 380 with me during JTerm. It's been extremely well reviewed over the years, and it's been a wonderful opportunity for me to develop a better understanding of students as I translate that understanding to things like medical school, recommendations, etc. So I hope you'll consider joining me for Bio 380. But again, I'm always happy to do advising meetings. And once you're a student in my class, I consider you an advising. So do not hesitate to reach out to me. You guys have been warriors. I'm so proud of my mighty ramblers this year in my Bio 282 class. You guys really brought that class in for a landing and did quite well on the final exam. So I laud you for your dedication despite the very trying circumstances. And thank you for being such a positive force in the class this year. You guys were incredibly proactive in working with me and reaching out to me and interacting with me in Zoom and in office hours. And I appreciate very much your guys' participation. I hope you and yours are well. Thank you for being in the course with me this year. Have a good day, my friends. Bye-bye.